So lesson 20, codominance. We're going to compare this to what we did last lesson, and then we're going to move further forward. Now that I've tried to remove some of the students that shouldn't have been in here, I'm just going to lock the classroom. Actually, I can do that. I can't remember how to do that. I'll do it later on. So what we started off by talking about was Mendel's P's. To do that, we talked about the idea of what did he discover about codominance. And he's essentially saying, yes.
Apparently Min, like from our class, was Aiden Augustine.
Well, that's what he gets for calling himself something wrong. So I kicked him out.
Yikes. That's his ass.
So Mendel, so essentially, I'm not gonna, I don't know how to let him back in because I did that before to Alex because she named herself something silly. So it's too late. Mendel, we're gonna keep talking about a lesson, so we're gonna try and push through. Mendel, what he discovered during his examples is he discovered essentially what he would call pure breeding, which is essentially when you've only got one type of gene showing up. That idea of pure breeding is called homozygous because ultimately you've only got that one gene, that one capital Y, that one lowercase y coming up over and over again. Okay? Now, when he bred two pure breeds, he would create a hybrid, which is why we got the word hybrid. And the word hybrid comes from this word heterogeneous, which is when you've got a different capital Y and a lowercase y. So that's something that's keep in mind that's pretty important for us. Carriers were the idea of hybrids can sometimes be known as carriers because they have these genes hidden in their DNA that they don't use, but they carry them and they can give them to other generations. And then we talked about this idea of partial dominance. Now, the first partial dominance we talked about before we ran out of time was incomplete dominance. Now, incomplete dominance is when you have a heterozygous organism, so an X or a Y, but instead both of them are dominant. So in this case, a red gene and a white gene, they're both dominant. So therefore, they make a pink colored because that's halfway between red and white. Now, a pink one can have two red, they can have two white, or they can have somewhere in between. Now, the next type we're going to talk about is something called codominance. Now, codominance, this is where a hybrid, in this case, a heterozygous.
Can we write this down in lesson 22 or lesson 21?
You should write this down in lesson 22.
Okay.
Codominance, this is where a hybrid organism receives both gene expressions. So they actually do both things at once. When you have a red and a white flower making a pink flower, that's a mixture. So the difference is this is both at once. So this is in blood types. In blood types, your body can make both A and B blood cells, or B, well, they're not really blood cells, they're more antigens, but still like antigens. And the idea is that it does both of them at the same time. So let's have a look at how they work for blood samples, because I think this is interesting. Let's just say you get the gene A and A. Well, that means, guess what? Your blood type A. Same thing occurs if you have B and B. That means your blood type B. Okay, those two are the easiest ones because it's like, well, it's the same. And O and O means your blood type O. Okay, that's the pretty simple one. Now, some of them, the ones with O, they're interesting. A and O, that means your blood type A. So the O gets ignored. Okay, if you've got A and O blood cells, O gets ignored. That sort of gives you the idea that the O blood type is recessive. It doesn't show up if you've got it doesn't show up if you've just got A.
Is that why, like,
yeah.
O is so compatible with a lot of different blood types, like when donating blood?
Yeah, that's why it is. It's not really recessive though, to be honest with you. O is sort of like, I think the absence, I think O from memory is the absence of antigens. So therefore they don't react. And because they don't have antigens, they don't react with anything. So they can be used for anything. But if you've got something that doesn't make antigens and something that does make antigens, it's just going to make A. Same with B and O. If you've got one that does make and one that doesn't make, then it's just going to make B. B is going to override it. It's kind of like, as I said, it's very similar to recessive, but it isn't actually recessive. The one that's interesting here, and the one that I want to take away from is this one here. AB, that's perhaps the most interesting. That's an example of codominance because you have genes A and B, but instead of being just A, or A being dominant and just making A, or B being dominant and just making B, you actually have a combination of A and B. So that makes it codominant. You show both of them at the same time. Now, I wanted to spend a little bit of time talking about a little bit of time as well talking about my experience with this and how I've got this. My blood type is A.
Wait, sorry, sir. Do we copy the table?
You don't need to copy the table. You can if you want. The main
Okay.
part is just what codominance is.
Okay.
My blood type is blood type A, and going back again to my parents are blood type O, and my father has blood type AB. Now, in order to be blood type O, my mother must be blood type OO, which means that I got an A from my father and O from my mother, which means that my blood type, my genetics, my genotype is actually AO. Now that manifests, shows up as me having blood type A. This shows like my father having blood type AB and my mother has blood type O. And yeah, this sort of shows up in a lot of things. The most common blood type in Australia is blood type A, so I am very, very common when it comes to these kinds of things. But in different countries, it's different. In some countries, it's O, in some countries, it's B, in some countries, it's AB. But yeah, essentially, the blood type, your genes that you get, you know, these genes, and this would be the genotype, and this would be the phenotype. This is how the genes manifest. This is what the genes are, and this is what they become. And AB is an example of codominance. Both the A gene and the B gene affect the phenotype. They affect what comes out at the end. And it's not a mixture. You don't get like an A-B mixture. You get both A and B showing up at the same time. That's what makes it codominant. Both showing up at the same time. So those are the two differences. You've got this idea of codominance, which means they both show up the same time, and you got this idea of going back, incomplete dominance, where it's sort of a little bit of both. The next thing I'm going to show you for today, which is actually going to be the last thing I'm going to show you, is I wanted to talk to you guys about. I've done a lot of these things where I'm predicting, like, you know, these family trees. We're going to spend a fair bit of time on Wednesday in this class talking about those family trees. But I wanted to start to talk about those family trees by talking about a Punnett square. Now, a Punnett square, you don't have to copy this Punnett square down because we're going to be making our own Punnett squares on the next slide. But I'm going to start off by talking about what a Punnett square is. Punnett squares are a way of visualizing the genotype of potential offspring of two organisms. So on the right here, we've got, back to Mendel's peas, we've got the yellow skin on on the casing is a dominant, whereas the green casing, or not yellow, is a recessive. So we've got this one, which is yellow, which is not yellow, not yellow. This is a homogeneous P, or in this case, a purebred, because it's only got one set of leaves, whereas this one here is a heterozygous. Homogeneous. Homozygous. Whereas this is a heterozygous, which means it's a hybrid. Now the idea of the Punnett square, and of course I can tell this is a hybrid because it's got a capital Y and a lowercase y, whereas this one, and that means it's got a dominant gene and a recessive gene. Now, how you would use this is that hybrid can either give a yellow, you can give a yellow, or a not yellow. Whereas the homogeneous one will either give a not yellow, or a not yellow. And you can then use these Punnett squares to then say, well, these are the genotypes. This is the phenotype. That's what shows up. Because it's got a dominant yellow, it shows up as yellow. Dominant yellow shows up as yellow. No dominant yellow, so it shows up as green. No dominant yellow, so it shows up as green. That's how you can use Punnett squares. Instead of trying to do those complicated tree diagrams that I showed you, Punnett squares make it a bit simpler. Now, this was a bit confusing because we were just looking at a Punnett square and trying to understand it. What's going to be a bit simpler is in the next slide where we create one. I should say that Reginald Punnett is the person 1875 to 1967. He was the one that sort of came up with these. He copied Mendel's ideas and sort of was like, he made it or copied them and figured out how to reproduce them. And so then he was the one that formalized these Punnett squares to working out things. So we're going to do some examples and we're going to give it a try. Let's look at, and we're going to do that on this slide. And this is going to be the last slide. So we're going to do complete dominance and incomplete dominance. We're going to wrap that up. So let's do this here. Let's look at complete dominance. To do a Punnett square, I usually go like, okay, it's not a very good square, but you get the idea. I'm going to go like this. And we're going to do, what do you guys want to do? Do you guys want to do eye color, or do you want to do right hand, or do you want to do tongue roll? Tongue roll. Eye color.
Tongue roll. Eye color. Wait, whoever eye color. I don't mind. I don't do.
Okay, well, let's do, you know what? I'm going to do tongue roll. The reason I'm going to do tongue roll is because if I do eye color, I hate doing eye color because then you get brown, and you can't tell if it's brown as in blue or brown as in green. We might do 2 examples. We'll do 2 examples for complete dominance, and then we'll look at incomplete dominance. We'll do that one, a little bit. So we'll do that one a little bit later. We're going to do tongue rolling. So for tongue rolling, we're going to say capital T, which is the dominant gene, is going to be you're a tongue roller. Okay? Okay, so now if you scored, instead of capital T, let's just say you have a lowercase t, that's going to be a non-tongue roller. Now we're going to do we're going to look at 2 people. Okay, and these two people are going to have children. One of these two people is going to be a tongue roll. Is going to be a non-tongue roller. So this person can't roll their tongue because they have two recessive genes, right? And then we're going to have a hybrid. Now here's my first question for the heterozygous hybrid. Can the heterozygous hybrid, can this person roll their tongue or not?
Yes.
Yes, this heterozygous person can roll their tongue, whereas the homozygous, can they roll their tongue? Yes or no? I'll ask the question again in case you missed it. Can the homozygous person, the lowercase t, can they roll their tongue?
No.
No, they can't. So I've decided just to pick randomly that the mum is the uppercase t, lowercase t, the dad is the lowercase t. How do their genes go? Well, the mum can either give an uppercase t or a lowercase t. Okay? The dad in this case can only give a lowercase t or a lowercase t. With the dad, I fill downwards. With the mum, I fill across. And now what I've done is I've created 4 different possible children. And those are the 4 possible children, the 4 possible genotypes their children could have. So now that we've got the 4 possible genotypes, let's grab a look. This top one here, top left, will this baby be a roller or a non-roller? This one here, roller or non-roller? If you don't want to say it out loud, you can put it in the chat. That's okay. Top one, capital T, lowercase t, roller or non-roller?
Voila.
Yeah, it's a roller. They can roll because they have a dominant T. What about this one here, top right? Roller or non-roller?
Roller.
Yeah, roller, because it's got a capital T. What about this one on the bottom left? Non-roller or roller?
Non-voila.
Non-roller, because it's a lowercase t. And last but not least, this one here, which has got lowercase t, that's going to be what?
Non.
Non-roller. Now, what's cool about this, what makes it useful, we can see there are 2 rollers and there are 2 non-rollers. So for this example, there is a 50% chance of children being rollers. There's a 50% chance of them being a roller. And that's, that's the same situation that occurs for me when we did yesterday or Wednesday, when we looked at blue eyes and brown eyes, was a 50% chance that my children will be blue eyes and all my children will be brown eyes because of this similar thing. The reason it's 50% as well is because we've got a homozygous and a heterozygous. Now you don't have to do it in that order. You could put the homozygous on the left, but yeah. What I want to do now is I want to do one more like this, But what we're going to do is we're going to look at we're going to do this one a lot faster. We're going to look at, in this case, a mother that is a capital T, right? She's a tongue roller because she's got a capital T. But this case, she is a homozygous because she has pure-blooded. She's come from a long line of tongue rollers. And again, the father this time is again a non-tongue roller. But again, this time he's still homozygous. So what happens if we mix two of these together? Well, we're going to fill the lowercase t's in first, going down. Then I'm going to fill
All of them would be rollers.
lowercase t going across. In this case, they are all capital T's. So 100% of children are rollers. Okay. Now you can get the only other way that you could do this is if you got two hybrids together. The two hybrids together actually do something interesting. Do you want me to do two hybrids together, or would you rather me just skip now and do the incomplete dominance? It's up to you guys. Do you guys understand this, or do you want me to do one more example before I move on? What do you guys think?
Wait, one more example of the complete.
Yeah. Do you want one more
Is that what
example?
you're asking?
Yeah. Do you want me to do one more example to complete, or do you want me to move on to the incomplete?
Move on.
Okay, I can move on. I'm going to move on. It doesn't matter because the actual advantage is the new one that I'm going to do for the incomplete is going to be very similar. We're going to look at, for the incomplete, we're going to look at R, which we're going to say is, we're going to look at the red, white flowers again. You're going to see R is going to be for red flowers, and W is going to lead to white flowers. We're going to look at, I'm going to go back for a second. We're going to look at just this section here. What happens when two hybrids meet? What happens when you have in this case, I'm not going to use red because I don't want to make it confusing. What happens when you have a red, white meeting a orange, a red, white? What happens when these two things meet? This one here is a homozygous. No, it's not. They're different. It's a heterozygous. Now, because this is incomplete dominance, right? They're both capital letters because they're both dominant. They can both be dominant, right? So for the first one, we're going to look at just the top. We're going to go down, red, red, white, white. And now for the purple, which is also, get this, is also a hybrid. So it's also heterozygous. That's going to have red, red, white, white. Again, going to the right for the purple, going up and down for the top one, the orange. We end up with a couple of different possibilities here. Can someone tell me what color is this flower in the top left? What's that going to be? What color is that flower?
Red.
Red.
And what's the chance of that flower showing up? What's the probability?
100%.
100%? No. Did you say 100? Sorry, Uriel, if you didn't say 100, I couldn't hear you very well. It is 50? No, David, it's not 50. There is only one combination of red. So in order to get a red flower, there is a 25% chance, or 1 in 4. Now, I think, Brendan Chang, remember when I kept saying to you, oh, there's a 1 in 4 chance that, you know.
Wait, what?
Yeah, I was talking to you on Wednesday. And
Oh yeah.
on Wednesday,
Yeah, what were you saying?
on Wednesday, I said there was a. So I'm going to mute everyone because there's background noise, we'll say. On Wednesday, I was like, you're confused with where did the 1/4 come from? This is where the 1/4 comes from. There are 4 possibilities. One of those is red. One of those for you is left hand. So that's why I said 1 out of 4.
I
Whereas
screenshotted the whole thing. I'm not trolling. I screenshotted everything.
if we go have a look at the white in the bottom corner, there's only one way to get a white flower. That means it's a 25% chance of getting white. It's 1 out of 4. But if I grab a look at this one over here, red, white, red, there is 2 ways to get pink. That means there is a 50% chance that you'll end up with a pink flower if you mix these 2 together. And that's how we can use Punnett squares to then predict. Notice how I can predict for each of these examples what the probability is. And so I can tell you, without even doing it, without even like seeing the children of these things, if a tongue roller, if a purebred tongue roller and a purebred non-tongue roller have children, all of their children will be tongue rollers. If I have,
Wait, so
yeah,
isn't 50% 2 over 4?
yes. Sorry, I got carried away. Whoop, and I changed the slide. That should be 2 over 4. There we go, fixed. Because then that makes up, yeah. So I can predict if I've got a hybrid, a heterozygous and a homozygous tongue rollers, then 50% of the children will be rollers, 50% will be non-rollers. This would be different if this was a purebred roller, because then pretty much all of them would be tongue rollers as well, because the tongue rolling gene would come over. So being able to construct these is something that, pardon me, being able to construct these is something that we're going to work on Wednesday. We're actually going to get some practice on it on Wednesday, and that's what we're going to work on. What I have set for you guys to do for homework is I would love you to work on, if I can just open this up, this, where is it? Where is it? Yeah. The style. Can you guys see that screen at the moment? Let me check. Yeah, you can see it. I'd love you guys to work on the style. I have added one new thing to your list of stuff to do, but it's in a new topic. And I'll show you what it looks like. Let me see. From a student's perspective, you should see these are the ones we've done before. I've put up glossary here, but there's no questions in that. It's just a bunch of words in case you need to look something up. But types of inheritance is the next one. Types of inheritance has got these questions we talked. That's the video that we watched this morning. We've got some questions about it. When Mendel bred yellow seeds, pea plants, and green seeds, their offsprings were, if you remember, they were all yellow. And then later on, they saw some weird things. We've got some examples of dominance. And down the bottom here, we've got this sort of stuff that we've been talking about. If you look at this one here, you have to type in, so you have to type into these. wonder if I can show you, because I couldn't do this before unless I was previous student. Pardon me. So for example, hey, oh, I already answered it. If these two are both lowercase, they're both the same. So I know that they are both homozygous, whereas these ones are both the same. So they are homozygous, and I'm not going to give you the rest of them. Hopefully you don't remember that one anyway. So I want you guys to work on these. Those questions there are pretty easy to do. There's some fill in the blanks based on when alleles for a genotype are the same, the genotype is called, and this is again homozygous versus heterozygous, stuff like that. It's just practicing that. Now I am going to be generous with you guys because I know that style has not been working very well. So because style hasn't been working very well, if it takes you a little bit longer to do this, like if it takes you all the way up till Friday to get this done, that's okay. But that's going to be the homework. I'm going to leave that there for you guys. It's now 3:06 actually, just on time. So have a good weekend. You've got some questions that I'd like you to do from this stuff, and I will see you guys on Wednesday, either in person or, you know,
Bye.
Bye.
Alright, see you soon.